But then we have this alpha-synuclein, which tends to aggregate in some cases. As you said, there is some genetic aspect of this disease. And what happens to the brain when this protein starts aggregating, accumulating, and where it's accumulating actually?
So Lewy bodies, these protein inclusions, have been known since 1912, when the German scientist, I don't know if he was a neuropathologist, I forget now, but his name was Friedrich Levy, working with Alois Alzheimer and the team in Munich. He identified in post-mortem tissue, brain tissue from Parkinson's patients, he found these inclusions that then a few years later were called Levy bodies. And at the time, we didn't have antibodies. We didn't know what the proteins were. We just knew it reacted in a specific way with some dyes that they were using at the time. But we knew those structures existed. But only in 1997, with antibodies, a colleague, Maria Grazia Spilantini, was able to find that those Lewy body structures stain positive for alpha-synuclein. So how this happens is still a mystery. What we know is that it seems to be an age-associated process. So Parkinson's disease usually, in most cases, doesn't appear early in life. It appears after 60, 65 years of age, although you have junior and early onset cases as well, in people around 30 years old, 35, 40. And you have juvenile forms that can happen in people that are 12, 15. Those are familial forms normally, those juvenile forms associated with mutations in other genes. So we think maybe the pathological mechanisms might be a little different. But in all the other forms, or all the other cases, or most, I should say most, not all, most cases have Lewy body pathology in the brain. So they have these protein inclusions. And we don't know why this happens. There's many possibilities. One is that some cells, for some reason, start overproducing the protein. And because this is a protein that normally is what we call intrinsically disordered protein, this means that under physiological conditions, the structure seems to be disordered. So the protein is not nicely folded as other proteins. It seems to be disordered. And this seems to be one important characteristic of the protein to enable it to perform different functions, to interact with different partners, different proteins. But what happens is that at some point, then this protein starts to misfold and to form aggregates, what we call aggregates, these protein clumps. Eventually, we think this is a continuous process, and you start with a small aggregate and somehow it starts to grow, and at some point it forms the big protein clump that we can then see under the microscope, and we recognize it looks like a Lewy body. But we think, and this is still debatable, but the evidence from all the studies in animal models and in cell models suggests that this protein aggregation can be a bad thing. It can cause toxicity. Now, there's colleagues now that are questioning this hypothesis and that say maybe what we have is not toxicity coming from the accumulation of the protein, but coming from the loss of normal function. Because if, instead of having the protein in its unfolded or partially folded form, if now you have the protein clumping in one part of the cell, that means you don't have enough protein available in other parts of the cell to perform the normal function. And so this is now an hypothesis that is being tested as well. So is this a proteinopenia disease where you have problems because you don't have enough functional protein, or is it a proteinopathy where you have problems because the protein is causing toxicity? And probably in the end it's going to be something in between. It's not just one or the other. I think it's going to be something in between. And we know from studies in animals and in cells that you can, under certain conditions, induce toxicity if you overexpress, if you cause this protein to aggregate. So we know this happens. We are just not very good still at inducing the formation of these inclusions, because in animal models, we and many others use transgenic mice or rats, even non-human primates have been used. And you've never seen, no one has ever seen something that you can say, oh, this is a Lewy body formed in the brain of a rat or a mouse or a monkey. No one has ever been able to do that. And we don't know if this is because these animals don't live long enough. We know, as I mentioned, the disease appears normally late in life. So it means that you need time for these structures to form and maybe to have all the components that make them look like a Lewy body. And in these animal models that live months or years, but not 60 years, maybe we just never reach the point where we can mimic this aspect of the disease. So there's a lot of open questions still, but we have evolved a lot in our understanding of the basic biology of the protein and cellular pathways that can be modulated to affect the aggregation and toxicity of alpha-synuclein.
Neuroscience and beyond. Know more, get inspired.